I hear that Kevin spoiled the surprise for you by letting you know that he wasn't going to be here today. But for those of you that are in my 314 class, I came to haunt you on a Monday as well as on Tuesdays and Thursdays. So I'm going to be talking to you today about cellular signaling, and I have no idea whether this volume is adjusted correctly, so give me a little help. Louder? Let's see if I can make this go up. Speech. Oh. Volume. Okay, this is not doing anything. Is that better? Is that better? Doesn't seem like it does. I have to talk into it, really. All right. How about putting it right here? Now can you hear me? Yeah? Kind of? No? Oh. All right, I'll try to talk louder. Okay. You guys okay up there? All right. So I was told that Kevin has already started talking to you a little bit about signaling. So what I'm going to do is pick up from where I think he left off. And so the first thing, obviously, about signaling is that it is the way that cells communicate with each other. And as you know, cells need to communicate with each other, whether they are single-celled organisms or whether they are cells that are in different parts of a multicellular organism. So you know that you have to have all of your cells function in coordination, so that if, for example, you were to hear a predator coming for you, your ears have picked up that information, but then that information relayed by your brain has to reach your different parts of your body, and particularly that information has to help you coordinate your responses so that your legs will carry you away from that region as fast as possible. So that obviously is a very simplistic situation where one part of your body has to coordinate with another, but pretty much everything that you do, your different cells have to be able to communicate with each other so that you function as a single organism instead of having different parts of your system doing different things that would not result in a coordinated response. So signaling is the way that multicellular organisms get all of their parts to function together. And the way that they do this is through sending molecules from one part of the body to another part of the body. So signals, you have, I'm sure, heard about the various kinds of hormones that are produced in one part of the body and that are sent through the bloodstream to other parts of the body, where they will tell the cells in that other part of the body what they should be doing, right? And so how do molecules actually do, how do they tell anything? Everything that you see happening inside of a living system depends on molecules being able to communicate with other molecules by way of interacting with them, right? So your signals are one kind of molecule, and you can have those signals be bound by another kind of molecules, which are these signal receptors. So receptors are typically proteins, and these receptors will bind to the signal molecules, and when they bind to the signal molecules, they help to pass on the message that those signals are bringing to the inside of the cell that needs to change whatever it's doing. Okay, so today what we'll be talking about is probably one kind of receptor particularly, or a couple of different kinds of receptors that belong in the same family. And overall, for this class, you will learn about several different kinds of receptors that are shown over here. One is the beta-adrenergic receptor, which is sensitive to epinephrine, also known as adrenaline. You'll learn about the insulin receptor, and that obviously is a receptor for insulin. And you will learn about the EGF receptor, and EGF stands for epidermal growth factor, and the epidermal growth factor receptor is the receptor that is responsive to EGF, which actually is a growth factor that sends messages to cells about whether they need to divide or not. Okay, so it's a growth factor, and it provides information about when a cell needs to divide. Insulin, as you know, is involved in signaling the uptake of glucose. So when you have large amounts of glucose circulating in your system, then insulin tells your cells to take that glucose up into their inside of themselves. And the beta-adrenergic receptor, which responds to epinephrine, aka adrenaline, is the receptor that is used in the fight or flight response and in several other responses, mainly your stress responses. So, of course, this is a receptor that, when it binds to epinephrine, which is made by your body in situations of stress, then that allows the cells, when they receive this signal, to mobilize energy stores. Meaning what? Meaning that if you had that predator coming after you, then your adrenaline soars, and when your adrenaline soars, then that goes and tells the cells in your body, you need to get out of here, and therefore you need to be mobilizing your energy stores in order to run away as fast as you can, right? So those are the kinds of things that we're going to be talking about, and in fact, I probably will not get to insulin receptor or epidermal growth factor receptor because there's so much to say about just the first class of receptors that we're going to talk about. So when you look at the business of what's going on when a signal comes to bind to a receptor, there's a very simple relationship between what happens when a signal binds to a receptor and what happens subsequently. So in this figure, what you see there's a signal, and that signal is going to be bound by a receptor. Signal, as you know, could be a hormone. It could be a variety of other things. There are different kinds of signals, but we're focusing for the most part on hormones for now. And so you've got the signal, and it's going to arrive at the cell, the target cell that it's going to take, that it's communicating with, and that target cell is going to have these receptors. And these receptors that we're talking about, all of the receptors that we're talking about for your purposes, are proteins that are integral membrane proteins. And so what they are proteins that are that stick out outside of the cell and stick out inside of the cell through the plasma membrane. Okay? So they are transmembrane proteins, and so you will have these receptors at the cell surface, and when the signal arrives at the cell surface, these proteins, the receptors, are going to bind to the signal, and when they bind to the signal, the receptor undergoes a conformational change. So you've got this protein that's embedded in the plasma membrane, and it's sticking out outside of the cell, and it's sticking down into the cytosol of the cell. And on the outside, when the signal arrives and it binds to the receptor, that causes the receptor protein to change shape. And that change in shape is conveyed into the interior of the cell. There is a change in the conformation of the protein. So the part of the receptor that's inside of the cell actually changes its shape as well. And because of that, that change in shape can be sensed by other proteins within the cell. So you've got other proteins within the cell that interact with the receptor that can sense that the receptor has changed its shape when the signal binds. Okay, so in that way, you can pass on the message from the receptor to the interior of the cell. So when you pass on the message into the interior of the cell, what you're doing is you're starting a process that we call signal transduction. So you see that word over there, transduction. Okay? And signal transduction refers to how the signal, which arrived in one form from outside the cell, is going to be changed into another form within the cell. Okay? So in other words, you had a signal, let's say you had epinephrine come bind to its receptor, right? So the receptor changes shape, and it passes that information on in the form of its shape change that tells the inside of the cell epinephrine has bound on the outside, right? So you've got on the interior, you've got other proteins that will now pick up that message, and they will convey that message to the rest of the cell so that the cell will now know to do other things, right, than it was doing before. And that process by which the signal changes form, where it came, you had the epinephrine molecule come to the surface of the cell, but now inside of the cell, other molecules are going to carry out that, carry that message onto the interior of the cell. That process is signal transduction. Okay, so now the signal is in a different form than it was when it was originally sent. So this signal transduction process actually also involves amplification of the message. So what's amplification of the message? So it is a process by which a single signal molecule arriving at the cell and binding to a receptor will actually, when that message is passed on inside of the cell, that message will be amplified because you will now have a large number of additional molecules that will send that message to the rest of the cell. And so a single signal molecule can have its message amplified within the cell. Okay? So you have the signal transduction process, you have the amplification, and what that does is it's actually spreading throughout the cell the information that that signal brought. So if that signal was epinephrine and its message to the cell, so to speak, is you need to mobilize your energy stores, then that information is going to be sent throughout the cell. And when it's sent throughout the cell, sorry, I'm not used to having the things hanging off of me. Okay, so when it's sent throughout the cell, you have the cell now has information that it needs to change what it's doing, and what those changes might be could be something like, you think, okay, what is the kind of change that a cell would undergo when it receives a signal? A change might be that it will now change the activity of some of its enzymes. So that could be kind of metabolic changes. So you will now have enzymes that are activated that were not active before, or in some cases you might have genes expressed that weren't expressed before, and so on. So these are the ways in which cells can respond. So these responses that you see at the bottom could be things like having enzymes activated or deactivated, or you could have genes expressed that weren't expressed before, and things like that. Those are the kinds of responses that result from a signal being bound to a receptor and that information being conveyed inside of the cells. Okay, so how is this achieved? You're thinking, okay, how do we do this? And the way that cells do this is by using what are called second messengers. Okay. So the first messenger coming to the cell was the signal that came from somewhere else, bound to a receptor, right? Okay, so once that receptor has bound the signal and passed on the message to the interior of the cell, I told you that the signal changes form, right? There's signal transduction. And so how it changes form is through the production of a variety of small molecules called second messengers. And these second messengers are able to diffuse throughout the cell and spread the message, so to speak, right? So you have a variety of small molecules that serve as second messengers. And they're second messengers because they are passing on the message that was brought by the first messenger, which was the thing that arrived at the cell and bound to the receptor. And so the second messengers are, some examples of second messengers are cyclic AMP and cyclic GMP. So these are small nucleotides, and these nucleotides can serve as second messengers. You can have ions, like calcium ions, that can serve as second messengers. And you can have molecules like inositol 1,4,5-trisphosphate, also known as IP3. And this molecule down here, diacylglycerol or DAG, that can serve as second messengers, as you'll see very shortly. Okay? So how are these second messengers produced? We're going to look at that in a little bit more detail in a few minutes. But what you have to remember is the original first messenger, your signal came, bound to the receptor. That message was passed on to the interior of the cell. There's a bunch of steps, various things going on inside the cell, and then you produce second messengers. The second messengers then spread out throughout the cell, and once they spread out throughout the cell, they're able to carry their message saying, you need to do something different. Okay? And you'll see what that is going to be different. Okay, so that's second messengers. So first what we're going to do is we're going to look at a class of receptors that are called 7-TM receptors. And don't write down everything that's on this table. This is just to show you that this class of receptors, the 7-TM receptors, is involved in a variety of functions within organisms. You can see that hormone action, hormone secretion, neurotransmission, chemotaxis, exocytosis, control of blood pressure, blah, blah, blah. There's a whole bunch of them. And all of these things are mediated through the action of these 7-TM receptors. Okay? And I'll tell you in a minute what these are. And the 7-TM receptors include that receptor that I told you about, the beta-adrenergic receptor, that is responsive to epinephrine. So let's take a look at what these guys look like, and you'll see in a minute. Oh, that wasn't the figure I was looking for. Oh, I keep clicking on the wrong one. Sorry. Okay, so this figure here is actually showing you what a 7-TM receptor looks like. Okay, so remember I told you that the receptor is a protein. It's a transmembrane protein, and it has a portion of the protein that sticks outside of the cell and a portion of the protein that sticks into the cytosol of the cell. Okay, so if you look at this figure, you see over here, there's the N-terminus of this protein. This is the lipid bilayer, obviously. This is the plasma membrane of the cell. You've got the N-terminus of this protein over here. So that's one end of the protein that's sticking out, and that's the outside of the cell. And then this protein goes back and forth through the membrane. So it is folded up and down. So it goes through the membrane, and the end of it, the C-terminus, is sticking into the cytosol over here. These cylinders over here actually are alpha helical regions of the protein, where the protein is anchored in the membrane, and if you count them up, you will find that there are 7 of them. Okay? So what that 7-TM stands for is 7 transmembrane helices. Okay? So these receptors, because of their structure, are called the 7 transmembrane helix receptors. Right? So they've got 7 transmembrane helices, and they have one end outside here and one end on the inside here. Okay, so these are the 7 transmembrane helix receptors. And this one of these 7 transmembrane helix receptors is the one that binds to epinephrine. So we'll look at that in a second. So when you look at this, you realize that the extracellular portion of this receptor is going to be the thing that binds to the signal. And then that's going to cause a change in the shape of this protein that is going to be conveyed here at the C-terminus. And at the C-terminus, some other protein will interact with this and will be able to sense that a signal has been bound on the outside, and then it'll be able to pass the message up. Okay, so what actually does this look like? Now I will show you this picture, which I've been clicking on all this time. And this picture is a close-up of what the 7TM receptors look like. And what you can see is that when you look at this, the way they're arranged in the membrane, they're not obviously flat out like that, but you see that they're arranged in such a way that you have a binding site, the ligand binding site shown over here, and you can see that there is a binding site for whatever that signal might be. In the case of the beta adrenergic receptor, which is shown over here, its binding site is here, and that's where the epinephrine would bind, right? Likewise, you see over here, here's another 7TM receptor, and in this case, this is a very interesting receptor because it is the it's a receptor that is known as rhodopsin, and it actually has a ligand binding site, and rhodopsin is involved in actually sensing light and is involved in vision. Okay? What we're going to focus on is this guy over here, and we'll take a look at what happens with that in a minute. So actually, before I move on, I have to point out that this is obviously the binding site for the primary messenger, your signal, right, that's coming in. So that would be epinephrine in this case. And as we already noted, epinephrine is the signal that's involved in your fight or flight response, right? So when you are under stress, then, you know, back in the day when that stress might have been something that was threatening you, that was a predator or maybe another person of your own species that you were trying to decide, should I fight this or should I run away from it? And that's what your response was to the secretion of epinephrine that then ended up in your bloodstream binding to your beta adrenergic receptor. Okay, so when you think about the binding of the primary messenger to your receptor, I told you that there is a conformational change that is conveyed to the C-terminus of this receptor. And then you want to think about, okay, what happens then and what proteins are then able to sense that change and then pass it on to the rest of the cell, right? And so it turns out that the 7TM receptors have a partner that helps them to sense the change when they bind to their signal. So you know that you had this receptor binding to the epinephrine on the outside, and on the inside, the conformational change happens, and that conformational change is sensed by a protein known as a Gprotein. Okay? And the reason it's called a Gprotein is because this is a protein that can bind guanine nucleotides. Specifically, it can bind GDP or it can bind GTP. Okay? So Gproteins are the thing that are going to sense at the tail of the beta adrenergic receptor. They can sense that there's been a change when the signal is bound on the outside of the cell. Okay? So Gproteins are going to pick this up, this change in the tail of the receptor, and this is a complicated ribbon diagram showing you what a Gprotein looks like, and here is the GDP bound to it. And what you want to remember is, let's see, that the Gprotein is actually made up of 3 separate polypeptide subunits, and those 3 separate polypeptide subunits are called alpha, beta, and gamma. Okay? So the alpha and the beta gamma are sort of associated with each other, and when there's no signal present, the alpha has GDP bound to it. So you have the situation where you have this G protein made up of 3 parts, alpha, beta, and gamma. And when there's no signal present, this alpha has GDP bound to it. And what you have to imagine here is you've got this plasma membrane, you've got the receptor that runs through the plasma membrane, it's ready to bind a signal, and nearby, this G protein is also anchored to the membrane by a lipid tail, right? So it's attached to the membrane by a lipid tail. And the binding of the signal then causes the change in the tail of the receptor, which is sensed by this G protein, which is near the tail of the receptor. Okay? So when the tail undergoes a change, the G protein that is near the tail of the receptor can sense that there is a change in the receptor itself, and now that change will cause this G protein to do something different than before. Yeah?
So the primary signal doesn't bind to the N-terminus and it's sticking out of the cell?
It does. Sort of the N-terminus is the part that makes the binding site. It's shown as, well, it's shown like a little thread sticking out, but when you saw that ribbon diagram thing where you saw the helices were all arranged in such a way that you could have a binding site.
Okay, so that's where the binding site
yeah,
was. Okay.
yeah,
And then it causes the
yeah.
transformation.
Yeah, so that's on the extracellular side, and then the C-terminus actually changes conformation when there's a signal bound on the extracellular side. And that C-terminal change is sensed by the G protein, which is also embedded in the membrane close by. So it's not embedded, I shouldn't say it's embedded, but it has a tail that allows it to be associated with the inside of the membrane, right? Yeah? So the binding of the signal is on the N-terminal side, and the C-terminal side also undergoes a conformational change when the signal binds, and that change is what is picked up by the G protein, which is nearby. Okay? Yeah. So, as I said, when there's no signal, the alpha subunit has GDP bound to it, and let's see where we go over here. These are not my figures, so I have to look at them a little carefully to figure out where. Let's see. That was what we looked at. Okay. Never mind. Maybe we've got a... All right, here we go. That's the figure I want. Okay. So here was your receptor. And you can see this yellow thing over here. That's the epinephrine bound over there. And there's a change in the cytosolic side of the receptor. And that's picked up by this G protein. And this G protein, when it senses that there's a change in the C-terminus of the receptor, that is, it senses that a signal has bound, what's going to happen is that change is going to induce a change in the alpha subunit such that it lets go of its GDP and picks up a GTP instead. Okay? So remember, when there's no signal, it has GDP bound. When the signal binds, that is conveyed to the alpha subunit of the G protein, and that is going to exchange its GDP for a GTP. Okay? So remember, what's happening here is it's swapping out a GDP for a GTP. It's not phosphorylating its GDP. Okay? So it's just letting go of the GDP, grabbing a GTP. Okay. So when it binds the GTP, there's yet another change that happens, and that change is that the alpha subunit with the GTP bound dissociates, it separates from the beta gamma. Okay? So where you had the 3 subunits interacting with each other and sitting over there with the GDP bound on the alpha, when the alpha swaps out its GDP for a GTP, the alpha separates from the beta gamma. Okay? Yeah?
So is GTP readily available around?
Yes. Yes. So these are all nucleotides that are available, that are soluble in the cytosol, right? Okay. Yeah.
I do have a question. So after GTP and the alpha part of the molecule is detached,
Uh-huh.
where does the beta and the gamma go?
The beta gamma is also, so there's actually two lipid tails. So the beta gamma is anchored by one lipid tail, the alpha is anchored by another lipid tail to the membrane. So they can separate, and actually, depending upon the response, the beta gamma may have a role to play later on, like it does, for example, when you have in the case of rhodopsin, when you, in the visual response, the beta gamma will actually go on and pass on some message to the rest of the cell. In this case, we're talking about the response to epinephrine, and the alpha is what we focus on, okay? But they remain associated with the membrane. They just separate out, okay? And with the, yeah?
Is it possible for the beta gamma to rebound with the alpha that has the GTP and cause it to regrab it?
You're getting ahead of the story. I will tell you when it comes back, okay? So when it has GTP bound to it, it doesn't actually associate with the beta gamma, because the binding of the GTP makes it such that the conformation changes and it dissociates, okay? But when it's finished its work, it'll go back and bind to the beta gamma. So wait for that part of the story to arrive, right? Okay, so we've got this GTP bound alpha subunit, and when the GTP is bound to the alpha subunit, as I told you, it undergoes conformational change, and now it is what we call an activated alpha subunit. And what does it do? Its lipid tail isn't shown here, but what it's doing is it's moving along the membrane, and it is going to now interact with another membrane protein, which happens to be an enzyme. Okay? So this membrane protein is adenylate cyclase, which is shown over here, and this activated alpha with the GTP bound is going to interact with the adenylate cyclase, and it's going to activate the adenylate cyclase. Okay? So everything so far, as you can see, has resulted from a signal binding to something, changing its conformation, and that is passed on to the Gprotein, which changes its conformation. It now binds GTP instead of GDP and separates out into the alpha and the beta gamma. Now the alpha with its GTP is going off over to the side, and it's going to interact with the adenylate cyclase, which is activated by the alpha subunit with the GTP bound to it. Okay, what happens when you activate the adenylate cyclase? Adenylate cyclase is an enzyme. Okay, so what is its function? Its function is to make cyclic AMP starting with ATP. Okay, so adenylate cyclase uses ATP as a substrate and creates this molecule, cyclic AMP, and you might remember from a few minutes ago that cyclic AMP was what? A second messenger, right? So it's a small molecule that is capable of diffusing through the cell and passing on the message of the signal that arrived at the cell. So this adenylate cyclase is responsible for making the cyclic AMP. And why is this important? This is important because the cyclic AMP can bind to a protein kinase, which is also an enzyme. And do you know what a kinase does? Phosphorylates other proteins, right? So protein kinases are enzymes that phosphorylate other proteins and change their activity by way of phosphorylating them, okay? So cyclic AMP is going to bind to protein kinase A and activate it so that it goes from an inactive form to an active form. And once you have protein kinase A activated, protein kinase A molecules can now go throughout the cell and they can phosphorylate various target proteins, right? So they can phosphorylate different target proteins and either activate or inactivate them as those proteins are depending on how they're regulated. Okay? So if you activate a protein kinase, that protein kinase can go and bring about a lot of changes in cells because they can activate or inactivate a variety of substrates. And those substrates are actually, the substrate of the protein kinase is other enzymes, other proteins, right? So those proteins will either, when they get phosphorylated, will either start being active or they may be inactivated depending upon the specific protein, right? Because the phosphorylation can act as either an on switch or an off switch depending upon the starting state of the protein. Okay, so we have, for example, if you think about the activities of enzymes that might be changed by phosphorylation by the protein kinase A, you can think of the effects that you might see. For example, in response to epinephrine, I said you want to mobilize your energy stores, right? So you're in a stress situation, you need glucose. You need energy. If you're going to run away from a predator, you're going to need to fuel your muscles. You need to have lots of glucose running through your system so that you can do what you need to do to get away from this stressful situation. Or if you're going to stay and fight, you still need that energy if you want to win that fight, right? So you're going to need that glucose. And so among the enzymes that could be changed by phosphorylation by protein kinase A are enzymes in your liver and in your muscles. And in your liver, for example, there are enzymes that synthesize glycogen, which you may recall is a storage form of energy, because glycogen is made up of strings of what molecule? Glucose, right? So you've got glucose stored up as glycogen, and you've got it in your muscles, you've got it in your liver, and if you want that glucose, you want to break that glycogen down, right? So what you can do then, how you're going to achieve this when your epinephrine binds to the receptor is that you pass the message on, you activate a protein kinase A. That protein kinase A is going to phosphorylate enzymes that are involved in glycogen breakdown and in the enzymes involved in glycogen synthesis. And the interesting thing is that the action of that phosphorylation, that is when protein kinase A phosphorylates these enzymes, the effect of phosphorylation on the enzymes that break down glycogen is to activate them, right? What do you need? You need to break down glycogen. So if you need to break down glycogen, you want to activate the enzymes that will break down glycogen. And the way you activate those enzymes is by phosphorylating them, and that phosphorylation is carried out by protein kinase A. Okay? And at the same time, the enzymes that synthesize glycogen are inhibited when they are phosphorylated. So you've got both those things happening. You want to stop glucose from being sequestered as glycogen. You don't want it to be tied up making glycogen. You want to release as much glucose as possible out into the system. And so you have the system where protein kinase A will phosphorylate and activate the enzymes that break down glycogen and will phosphorylate and inhibit the enzymes that synthesize glycogen. Make sense? Okay. All right, so we've got this whole thing playing out. You've got signal, the first messenger, binding to receptor. Receptor changes shape, affects the G protein such that it swaps out its GDP for GTP and separates out into alpha and beta gamma. This alpha goes and activates adenylate cyclase. Adenylate cyclase starts making a lot of cyclic AMP, and that is going to activate protein kinase A. Protein kinase A is going on to activate the enzymes of glycogen breakdown, and now you will have mobilized glucose for you to be able to run away from the predator, right? So that's your whole pathway there. So then, I'm sure that you're thinking, there's a problem with this system, right? So once this thing binds over here, and it sets off this whole series of events. You don't want to deplete every last molecule of glycogen left in your system. So how does this whole thing turn off, right? Because you just turned on a response, but you don't want it to run forever. You don't want to be in a stressed state forever. You want to have that glucose. You want to get away from the predator, but then you want things to calm down in your system, right? So you have to think about how this might be inactivated. That is, you activated a system that set in motion a whole series of events within your cells. Now you want to be able to switch those off. So how do we do this? Let's see if I can find the correct. Oh, that was just the thing that we talked about, so let's not worry about that one. All right. Let's take a look at this. Okay. So it turns out that you can reset the system, that is, turn off this response in several different ways. Okay? So one way that you can turn the system off is actually built into the Gprotein. So how do you build this into the Gprotein? We know that activated alpha subunit has got GTP bound to it, right? And you remember that when it was in its inactive form, it had GDP bound to it, right? So what you're going to have to do is to turn this GTP back to GDP to inactivate that alpha subunit. And it turns out that the alpha subunit actually has the ability to hydrolyze GTP to GDP. So here's the alpha subunit. It swaps its GDP for a GTP and becomes activated, right? And then it's going to go off and activate the adenylate cyclase over here so that it'll make cyclic AMP. But the alpha subunit, once it's bound to GTP, actually in a minute or two starts hydrolyzing it. Okay, so it starts breaking down the GTP, and pretty soon there is the alpha subunit. It's got GDP bound to it. Once it has GDP bound to it, it's no longer able to activate the adenylate cyclase, right? And once it has GDP associated with it, it can now go back to its old friend beta gamma and form the trimeric complex that was the inactive Gprotein, right? Okay, so you have a way for the alpha subunit to hydrolyze the GTP bound to it, return to its GDP bound form, and then reassociate with the beta gamma so that now it is back in the inactive form. Okay. So it's actually the alpha subunit is turning itself off by hydrolyzing the GTP to GDP. Okay. There's another way that you can control this or turn off the response, right? Because one way to turn off the response is to render the alpha subunit inactive by having the GTP hydrolyzed to GDP. And so, is what?
Question?
Yeah, sorry.
So does this happen even if the receptor is still attached to the surface membrane, or does the receptor have to be taken off for this to happen?
No. So the hydrolysis of the GTP is going to happen independent of the other steps, but I'm going to talk about those in a minute, okay? So there's other ways in which your response can be damped down, okay? And one of them would be obviously the release of the signal by the receptor, right? So you know that the epinephrine, for example, came and bound to this receptor on the extracellular side, and that is not a permanent binding, right? So you can release that signal from the receptor, and that would be the way that you could turn it off. Okay, so signal termination. So you see over here, for example, here is that receptor with the epinephrine bound, and you can have the epinephrine dissociate, and then that receptor goes back to its normal state, so it's not going to interact with and activate the G protein, right? So that's another way that you can turn that signal off. A third way to turn the signal off is actually where you have an enzyme called a receptor kinase, and that receptor kinase can actually phosphorylate the C-terminal tail of this receptor. And when that C-terminal tail is phosphorylated, a protein called beta-arrestin comes along and binds to that C-terminal portion that normally would have interacted with the G protein, right? So basically you're blocking that portion where it could have interacted with the G protein, and so it can't pass that signal on. Does that make sense? Okay. So there are 3 different ways here that show you how you can turn this off. You've got the alpha subunit hydrolyzing GTP to GDP. You've got the receptor letting go of the signal. You have the beta-arrestin, which can effectively block that G protein interacting region. But there's yet one more place that you can stop these things from, stop this process from happening. And that's not the one I was looking for, if I could find it here. There is an enzyme that is involved in breaking down the cyclic AMP. Okay? So you know that you made the second messenger, and that was spreading out and activating protein kinase A. So another way that you could regulate that response is by having that second messenger mopped up, right? You remove the second messenger, you don't activate protein kinase A. So there's an enzyme called phosphodiesterase, and phosphodiesterase, which I don't see a link to here. Does anyone see it? All right, it's not on there. Phosphodiesterase is an enzyme that can break down cyclic AMP. Just like adenylate cyclase makes cyclic AMP, phosphodiesterase is going to break cyclic AMP down. And so what you do is you remove all of that cyclic AMP from circulation, and now you have another way that you can stop that response from going on and on and on, and that is to remove that cyclic AMP. Is that making sense? Okay. All right. So we talked about cyclic AMP, and there's one other kind of response that you can have with the 7-TM receptors, and the 7-TM receptors, in this case, a different 7-TM receptor called an angiotensin receptor uses a different system to activate it. And the system that it uses involves 2 other second messengers, and those second messengers are those molecules that you saw before, IP3 and diacylglycerol, or DAG. Okay? And in this case, the angiotensin receptor binds to angiotensin, and angiotensin is a molecule that regulates blood pressure, right? So it's your blood pressure that is regulated by angiotensin, and angiotensin also binds to a 7-TM receptor, a different 7-TM receptor. Okay? So it's got its own receptor, that is the angiotensin receptor, and how it works is in the same way. It activates a G protein, and that G protein activates a different enzyme, not adenylate cyclase, but it activates an enzyme called phospholipase C that breaks down this molecule that is membrane associated into these 2 molecules, DAG and IP3, and DAG and IP3 are the second messengers for this pathway that is going to activate a different set of protein kinases. Okay? So you can use these molecules to activate a different protein kinase. We're out of time, and Kevin will pick up next time and explain how that all works.